The boundary between the somatic and the germline is not merely a biological distinction, it is a moral event horizon. In the somatic cell, the lung, the liver, the skin, the DNA is a local script, a set of instructions relevant only to the immediate life of the organism. When that organism dies, the script dies with it, its mutations and edits dissolving into the entropy of the earth. But the germline, the sperm, the egg, the zygote, operates on a timeline that transcends the individual. It is the river of continuity that flows from the deep past into the unknown future. To edit the DNA of a single-cell human embryo is to introduce a change that will be replicated in every cell of the developing fetus, from the neurons of the brain to the beating cells of the heart, and crucially, into the germ cells of that new individual. Before we move on, if you like learning this way, you can quietly hit subscribe so you don't lose this channel later. It is an edit that will be passed to their children, and their children's children, becoming a permanent feature of the human genetic heritage. For years, this possibility hung over the field like a heavy, suspended storm cloud, technically feasible, yet ethically electrified. The consensus among the global scientific community was a fragile, unspoken pact. We will refine the tools, we will cure the sick, but we will not touch the embryo. We will not design the future. In November 2018, that pact was shattered, not with a whisper, but with a declaration that reverberated through the halls of the Second International Summit on Human Genome Editing in Hong Kong. A Chinese biophysicist named He Jiankui took the stage, and in a calm, almost detached manner, revealed that he had already crossed the line. He had used CRISPR-Cas9 to edit the embryos of twin girls, known to the world by the pseudonyms Lulu and Nana. His target was the CCR5 gene, a protein on the surface of white blood cells that acts as a doorway for the HIV virus. By disabling this gene, he intended to bestow upon these children a genetic resistance to HIV, mimicking a rare natural mutation found in a small percentage of Northern Europeans. The revelation was met with a stunned, suffocating silence, followed by a tidal wave of condemnation. But stripped of the media storm, the scientific reality of the experiment revealed the profound immaturity of the technology for such a purpose. The data presented was messy. The editing was not perfect. In one of the twins, the edit was mosaic, meaning that some cells carried the modification while others remained wild type, creating a patchwork organism with uncertain protection. Furthermore, the edits created were not the precise natural mutation he sought to mimic, but novel, unstudied deletions whose long-term physiological effects were entirely unknown. The CCR5 gene is not vestigial. It plays a complex role in the immune system and brain function. To delete it based on a hypothesis in a healthy embryo was a gamble with a human life. The He Jiankui affair served as a jarring wake-up call, a demonstration that the raw power of Cas9, the ability to break DNA, was a blunt instrument, perhaps too blunt for the delicate inception of life. It highlighted the perils of the double strand break. When Cas9 severs the double helix, it is an act of molecular violence. As we have seen, the cell screams in panic, and the repair pathways are unpredictable. For a therapy in an adult patient, a certain level of chaos is acceptable. If a small percentage of blood cells fail to edit correctly, the patient still benefits from the millions that do. But in an embryo, there is no margin for error. A single off-target cut, a single unwanted deletion, becomes a foundational flaw in the architecture of the resulting human. This realization drove the field to pivot, to seek a new generation of tools that could edit the code without breaking the bone. The quest became one of elegance. How do we rewrite the genetic letters without cutting the page? From this pressure cooker of necessity emerged base editing, a technology developed largely in the laboratory of David Liu at the Broad Institute. If CRISPR-Cas9 is a pair of scissors, base editing is a pencil with an eraser. Liu and his team realized that the true danger lay in the double strand break. They asked, can we chemically convert one DNA base into another directly, without snapping the backbone? To achieve this, they engineered a dead version of Cas9, dCas9. This protein still possessed the homing ability. Guided by RNA, it could search the genome and clamp onto a specific sequence. But its cutting blades were deactivated. It was a sterile missile, capable of finding the target but incapable of destroying it. To this silent guide, they fused a second enzyme, a deaminase. In nature, deaminases are enzymes that perform chemistry on nucleotide bases, often as part of immune defense or RNA editing. By tethering a cytidine deaminase to the dCas9, the researchers created a machine that could grab the DNA, unwind the helix to expose the single strands, and then perform a subtle chemical reaction on a specific cytosine C. It removes an amino group, chemically transforming the cytosine into uracil, U. In the language of DNA, uracil reads like thymine, T. When the cell replicates this strand, it sees the U and places an adenine, A, across from it. In the next round of replication, the A pairs with a T. The result is a clean conversion of a CG base pair to a Tia base pair. No double strand break. No activation of the panic repair pathways. No chaotic insertions or deletions. Just a precise single-letter substitution. This was a quantum leap in refinement. A significant portion of human genetic diseases, around 30%, are caused by point mutations, single-letter errors where a C is in the place of a T or a G is in the place of an A. Base editors offered a way to correct these errors with a fidelity that standard CRISPR could never achieve. Later, the team developed adenine base editors, capable of converting AT pairs to GC pairs, a feat that had no natural enzyme precedent and required the forced evolution of a bacterial protein in the lab to achieve. Suddenly, the vast majority of pathogenic point mutations were within reach of these chemical pens. The narrative of the field shifted from cutting and disrupting to correcting and rewriting. But even base editing had its limits. It could only change certain letters to certain other letters, purines to purines, or pyrimidines to pyrimidines. It could not easily insert new text or delete specific stretches. It was a spell checker that could fix typos but could not rewrite a sentence. The hunger for total control remained. The ultimate goal was a search and replace function for the genome, a tool that could find any sequence and replace it with any other sequence of our choosing, regardless of length or composition, and do so without the risks of double-strand breaks. In 2019, the same laboratory that brought us base editing unveiled prime editing, a technology of such mechanical complexity and ingenuity that it effectively turned the Cas9 complex into a biological word processor. Prime editing involves a fusion of the nicked Cas9, which cuts only one strand of the DNA, avoiding the lethal double break, and an enzyme called reverse transcriptase. Reverse transcriptase is a retroviral engine. It is the enzyme viruses like HIV use to copy their RNA genomes back into DNA. In the prime editing system, the guide RNA is extended into a long, complex structure called a pegRNA, prime editing guide RNA. This pegRNA plays a dual role. Its first section guides the Cas9 to the target site, just like standard CRISPR. But its tail contains a template, a blueprint of the desired edit. The mechanism is a ballet of molecular interactions. The Cas9 nicks one strand of the DNA. The reverse transcriptase grabs the dangling end of the cut DNA and uses the tail of the pegRNA as a template to extend it, literally writing the new genetic information directly onto the end of the chromosome. The cell then incorporates this newly written flap into the helix, overriding the old sequence. Prime editing is versatile. It can change any letter to any other letter. It can insert clean segments of DNA, and it can perform precise deletions, all without severing the chromosome. It is slower, larger, and more cumbersome than the original Cas9, but its precision is unparalleled. It represents the maturation of synthetic biology from a disruptive force to a constructive art. While these advanced editors were being forged in the fires of academic research, the original CRISPR-Cas9 was finally meeting its destiny in the clinic. The shadow of the He Jiankui affair had cooled the fervor for germline editing, redirecting the world's attention back to the somatic patients who were suffering now. The most poignant chapter of this saga began to unfold in the blood of a woman named Victoria Gray. Gray lived with Sickle Cell Disease, a brutal genetic condition caused by a single mutation in the hemoglobin gene. This mutation causes red blood cells to deform into rigid crescents that clog capillaries, causing excruciating pain, organ damage, and shortened lifespans. For her entire life, her own blood had been her enemy. In 2019, Gray became the first patient in the United States to be treated with a CRISPR-based therapy for sickle cell disease. Doctors extracted her bone marrow stem cells, the mothers of all blood cells. In a specialized manufacturing facility, scientists used CRISPR-Cas9 to edit these cells. They did not fix the sickle mutation directly. Instead, they employed the BCL11A strategy, breaking the break to reactivate fetal hemoglobin. Millions of her stem cells were edited, checked for safety, and then infused back into her veins. It was a rebirth. The edited cells found their way back to her bone marrow, engraved themselves into the hollows of her bones, and began to produce a new kind of blood. The results were nothing short of miraculous. Within months, her system was flooded with fetal hemoglobin. The sickle cells disappeared. The pain crises which had plagued her for decades vanished. She had been effectively cured by a molecular machine derived from a bacterium, engineered by humans, and reintroduced into her own body. This was not a theoretical success, it was a woman reclaiming her life. Her success and the success of others in similar trials marked the transition of CRISPR from a promising technology to a medicine. It validated the decades of basic research, the sequencing of salt marsh microbes, the crystallization of proteins, and the late nights in tissue culture rooms. However, as the celebrations of somatic success rang out, the field remained acutely aware of the disparity of access. Victoria Gray's treatment was a bespoke, high-tech procedure costing millions of dollars, requiring advanced hospital infrastructure and chemotherapy to clear the marrow. It is a cure for the few. The vast majority of sickle cell patients live in sub-Saharan Africa and India, far from the sterile clean rooms of Western gene therapy centers. The revolution was successful, but it was not yet equitable. This realization has spawned a new wave of research focused on in vivo delivery, finding ways to inject the CRISPR machinery directly into the patient, perhaps as a simple infusion or a shot, bypassing the need for complex cell transplantation. The narrative of the edit is thus one of constant oscillation between the expansive potential of the future and the granular constraints of the present. We have moved from the blunt trauma of the double-strand break to the elegant calligraphy of prime editing. We have looked into the abyss of the germline and stepped back, choosing instead to focus on the suffering of the living. But the tools continue to sharpen. The library of Cas enzymes grows. The understanding of the genome deepens. We are building a relationship with our own biology that is no longer passive. As we stand in this current moment, the synthetic biology revolution is evolving into a synthetic ecosystem. It is not just about editing humans. The same tools are being turned toward the environment. Researchers are engineering coral to withstand warming oceans, editing the genes of the American chestnut tree to resist the blight that wiped it out a century ago, and designing bacteria that can eat plastic or sense toxins in the soil. The rise of the edit is becoming the rise of stewardship, a tentative, hopeful, yet dangerous idea that we can use these tools to repair not just our broken genes, but our broken planet. But every edit is a question. When we change the chestnut tree, do we understand the fungal ecosystem that depends on its rotting wood? When we release a gene drive to suppress mosquitoes, do we know what will fill the ecological void? The precision of the tool does not guarantee the wisdom of the user. The scientific script is being written in real time, and the ink is wet. The tension between our ability to manipulate the world and our ability to understand the consequences of that manipulation is the defining drama of our age. In the final movements of this exploration, we must look at the convergence of CRISPR with the other great titan of modern science, artificial intelligence. The marriage of machine learning and gene editing promises to accelerate the revolution beyond the speed of human thought. Algorithms are now being trained to predict the off-target effects of guide RNAs, to design new Cas proteins that have never existed in nature, and to simulate the outcomes of edits before they are ever performed. We are entering an era where the code of life is deciphered by the code of silicon, a feedback loop of intelligence that may unlock the final secrets of the genome.